Welcome to Roadmap to Residency Evidence-Based Applications. Hello everyone, I am Abhiraz. Welcome to our Club 250 Matchmaker sessions. Here in this video, we will be discussing some of the important topics from biochemistry, the oxidative phosphorylation, the TCA cycle and all. So talking about electron transport chain and oxidative phosphorylation. So this is from your first aid only. So the NADH electrons are transferred to complex one. Okay, so here's the complex one. So NADH changes to NAD plus. So we will discuss it all in steps in the further slides. So just a review here. And then the FADH2 electrons are transferred to complex 2. Here we can see by the help of the enzyme succeed dehydrogenase. The passage of electrons results in the formation of a proton gradient that, coupled to oxidative phosphorylation, drives the ATP production. The ATP hydrolysis can be coupled to energetically unfavorable reactions. The uncoupling reactions, found in brown fat, which has more mitochondria than white fat, produce heat by increasing the mitochondrial membrane permeability and decreasing the proton gradient. The ATP synthesis stops, but electron transport continues. So one NADS will yield 2.5 ATP and one FADS2 will yield 1.5 ATP. The NADS electrons from glycolysis enter the mitochondria via the malate aspartate or glycerol 3-phosphate shuttle. We will discuss this shuttle in the future slides. The aerobic metabolism of one glucose molecule produces 32 net ATP via the malate aspartate shuttle, 38 ATP via glycerol 3-phosphate shuttle. The anaerobic glycolysis produces only 2 net ATP per glucose molecule. The aspirin overdose can also cause uncoupling of oxidative phosphorylation, resulting in hyperthermia. So, this aspirin overdose is causing hyperthermia due to effect where? So, in the mitochondria. A very, very important question, okay, that you also asked in your self-exams. So, this is the summary of electron transport chain and oxidative phosphorylation, which you need to remember at last, but we will be discussing this in detail. So the TCA cycle, also called as a tricarboxylic acid cycle, Krebs cycle, or citric acid cycle, is a metabolic pathway which converts acetyl-CoA to CO2 or carbon dioxide. It derives energy from reactions, and all reactions occur in mitochondria. What does it do? So it produces NADH and FADH2, which is then sent to electron transport chain to produce ATP. It produces GTP and it produces carbon dioxide. So the TCS cycle, okay, so whenever you have acetyl-CoA, it combines with oxaloacetate to form citrate. This citrate is converted to isocitrate, which yields CO2 and NADH to go and form alpha-keto glutarate. Now, this alpha-keto glutarate again produces CO2 and NADH. So we have two CO2 molecules and two NADH molecules, and they form succinyl-CoA. This succinyl-CoA forms the GTP, okay? So now we have from two CO2, two NADH, one GTP. And this succinyl-CoA forms succinate, and now this succinate can further disintegrate to form FADH2 and fumarate. So what we have by far is CO2, NADH, 2 CO2, 2 NADH, 1 GTP, 1 FADH2. And again, that goes to form in a malate molecule, which again forms the NADH. So we have the 3 NADH molecules here. And again, this malate forms oxaloacetate and the cycle continues. And this is called as the TCA cycle. So the citrate synthesis. Citrate is a 6-carbon structure. So oxaloacetate combines with acetyl-CoA. So the 4-carbon structure combines with 2-carbon structure to form citrate. And this reaction, or the citrate synthesis, or the citrate synthase enzyme, is inhibited by ATP. So if there is abundant amount of ATP, the reaction will not progress forward. The fasting state. So in fasting state, the oxaloacetate is used for gluconeogenesis. If there is decreased oxaloacetate for the DCA cycle, and acetyl-CoA or the fatty acids form the ketone bodies. Talking about the compound isocitrate, which is the isomer of citrate, and the enzyme aconitase converts citrate to isocitrate. It forms intermediate or cis-aconitate, then isocitrate, and it is inhibited by fluoroacetate, which is present in rat poison. So talking about alpha-ketoglutarate. So alpha-ketoglutarate is a rate-limiting step of TCA cycle, very important statement, and it is inhibited by ATP and NADH. So if you have abundant amount of energy, ATP and NADH, then the reaction will not progress. So that's why it is called as a rate-limiting step of TCA cycle, and it is activated by ADP and calcium. Talking about succinyl-CoA, so alpha-ketoglutarate dehydrogenase complex is similar to pyruvate dehydrogenase complex, and it has cofactors like thiamine, CoA, NAD, FADS, and lipoic acid. So the alpha-ketoglutarate, okay, it combines with CoA to form succinyl-CoA by yielding CO2 and NADH. The enzyme here is alpha-ketoglutarate dehydrogenase, which is activated by calcium, and it is inhibited by succinyl-CoA. So if there is abundant amount of succinyl-CoA, it will again give negative feedback. And also if there is abundant amount of energy or NADH, then it will again give a negative feedback to alpha-ketoglutarate dehydrogenase, and there will be no formation of succinyl-CoA further. Talking about fumarate. So fumarate, so the succinate dehydrogenase, so unique enzyme, which is embedded in the mitochondrial membrane. It functions as complex 2 in the electron transport, and it is also produced in several other pathways like urea cycle, purine synthesis, and in amino acid breakdown. So from the succinate, by the enzyme succinate dehydrogenase, which is present in complex 2, we go on to form fumarate, okay, by production of FADH2, and this FADH2 goes to the electron transport. Now, malate and oxaloacetate, okay, now the NAD as such, and so we have the oxaloacetate, which by the enzyme, okay, malate dehydrogenase, okay, this is a bidirectional reaction. So either oxaloacetate can form malate or malate can form oxaloacetate. And again, there is fumarase, okay, from fumarate to malate and all. So talking about the malate shuttle. So malate shuttles molecules from cytosol towards the mitochondria. The key points is that malate can cross mitochondrial membrane, so it is a transporter. NADH and oxaloacetate cannot cross the mitochondrial membrane. So there are two key uses of malate shuttle: transfer of NADH into the mitochondria and transfer of oxaloacetate out of the mitochondria. So number one is the transfer of NADH. So here we can see that the aspartate uh, is equal to oxaloacetate. It forms oxaloacetate by the enzyme alpha-ketoglutarate, and then again the oxaloacetate forms the malate, okay, by the loss of electrons, right? And again, that's how this malate can cross the mitochondrial membrane, okay, and then the malate comes inside the mitochondria, okay, inside the mitochondrial matrix, and then again it can form oxaloacetate and aspartate. Use number 2 is the transfer of oxaloacetate so the oxaloacetate can either go into gluconeogenesis, or it can go into form malate, okay? Or we can say that from the mitochondrial matrix, the oxaloacetate gets converted into malate, okay? So in the mitochondrial matrix, the oxaloacetate here is getting converted into malate. This malate can cross the cytosol, and again this malate can go on to form oxaloacetate. So this is how you have transferred oxaloacetate from inside the mitochondria towards the outer cytosol. So TCA intermediates, okay. So we have the oxaloacetate forming citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, and succinate, which again forms fumarate, malate, and all, okay. So succinyl-CoA. So the TCA cycle can go to form succinyl-CoA. The succinyl-CoA can again go towards the TCA cycle. The methylmalonyl-CoA can form succinyl-CoA, which can again go to heme synthesis. So here we have a 35-year-old woman who presents to her primary care physician with complaints of fatigue, muscle weakness, and intermittent dizziness over the past few months. She reports feeling generally unwell and has noticed a decrease in her exercise tolerance. She denies any significant medical history or recent illnesses. On physical examination, the patient appears pale and fatigued. Neurological examination is unremarkable. Lab tests reveal elevated levels of lactate and pyruvate in her blood. Arterial blood gas analysis shows a normal PS with an elevated serum lactate to pyruvate ratio. Further metabolic testing confirms the deficiency of an enzyme involved in the TCA cycle. Additionally, muscle biopsy demonstrates ragged red fibers on histological examination. So which of the following enzymes is most likely deficient in this patient? So citrate synthase, isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase, succinate dehydrogenase, and malate dehydrogenase. So this patient's presentation, including symptoms of fatigue, muscle weakness, and intermittent dizziness, along with lab findings of elevated lactate and pyruvate, suggests a dysfunction in the tricarboxylic acid cycle, also known as the citric acid or Krebs cycle. Now, the TCA cycle responsible for generating energy in the form of ATP through the oxidation of acetyl-CoA. derived from carbohydrates, fats, and proteins. Additionally, the presence of ragged fibers in muscle biopsy is indicative of mitochondrial dysfunction, further supporting the suspicion of a mitochondrial disorder. The enzyme most likely deficient in this patient is alpha-ketoglutarate dehydrogenase. Thank you for watching. Keep studying hard.